Dr. Baliga here. This podcast is on the race to a vaccine for COVID-19. Today, Moderna, the company, has announced that FDA has completed its review of this company's investigational new drug application, that is IND application, for its novel coronavirus vaccine candidate, messenger RNA 1273. The FDA will now allow the vaccine to proceed to a phase 2 study, which is expected to begin shortly. Other companies which are neck and neck in this race are Pfizer, J&J, and Merck. The Biomedical Advanced Research and Development Authority, BARDA, is working with counterparts across the government to review potential vaccines, treatments, and diagnostics from across the public and private sectors to identify promising candidates that could be developed to detect, protect against, or treat people with coronavirus infection. It's working with the company Regeneron to leverage their partnership agreement to develop multiple monoclonal antibodies that individually or in combination could be used to treat this emerging coronavirus. Regeneron's monoclonal antibody discovery platform called Baliga was used to develop a promising investigational 3 antibody therapeutic which was deployed to treat Ebola in the most recent outbreak in the Democratic Republic of Congo, and an investigational 2 antibody therapeutic to treat MERS. The technology shortened multiple aspects of product development timeline for therapeutics to treat MERS and Ebola from years to months. Regeneron is working to utilize these 3 monoclonal antibodies produced by a single clone of cells or a cell line with identical antibody molecules which will bind to certain proteins of a virus, reducing the ability of COVID-19 virus to infect human cells. BARDA is working with Janssen to leverage their Ebola Zika HIV vaccine platform to expedite the development of vaccines that protects against the SARS-CoV-2 virus. Janssen's investigational Ebola vaccine was developed by the same approach with BARDA support, and that vaccine is being used in the Democratic Republic of Congo as a part of the current Ebola outbreak response. Additionally, BARDA and Janssen are working together to develop therapies for coronavirus infections. Janssen will conduct high-throughput screening on thousands of potential antiviral compounds in order to identify medicines that could safely and effectively be used to reduce the severity of illness and treat COVID-19 infection, as well as identify compounds that have antiviral activity against SARS-CoV-2 as an initial step in developing new therapies. These products include those in the development to treat and prevent MERS or SARS, which are caused by coronaviruses also related to COVID-19. BARDA is also working with Sanofi Pasteur to leverage a licensed recombinant influenza vaccine platform to produce a recombinant SARS-CoV-2 vaccine candidate. The technology produces an exact genetic match to proteins of the virus. DNA encoding proteins will be combined with DNA from a virus harmless to humans and used to rapidly produce large quantities of antigen which stimulate the immune system to protect against the virus. The antigens will be separated and collected from these cells and purified to create working stocks of vaccine for advanced development. The National Institute of Allergy and Infectious Disease Vaccine Research Center, VRC, collaborated with Moderna on the development of a vaccine candidate using mRNA or messenger RNA vaccine platform containing the gene that expresses VRC-designed spike protein of SARS-CoV-2. The National Institute of Allergies and Infectious Diseases, Rocky Mountain Laboratories, RML scientists are collaborating with Oxford University investigators to develop a chimpanzee adenovirus vector vaccine candidate against SARS-CoV-2. In addition, they have partnered with CureVac on an mRNA vaccine candidate. The Rocky Mountain Laboratory investigators have also launched a collaboration with the University of Washington and have begun an early stage testing of an RNA vaccine candidate of SARS-CoV-2. In addition, the National Institute of Allergy and Infectious Disease supports scientists at the Baylor College of Medicine and their collaborators and are evaluating an experimental SARS-CoV-2 recombinant protein vaccine to determine if it also provides protection against SARS-CoV-2. The National Institute of Allergies and Infectious Diseases is also exploring additional collaborations and industry partners on other vaccine concepts. It's also supporting the development of standardized assays and animal models that will be utilized to evaluate vaccine candidates. According to a Nature article, there are at least 8 types being tried against the coronavirus, and they rely on different viruses or viral parts. There are an array of vaccines. The virus vaccines include inactivated and weakened viruses vaccines. The viral vector approach includes replicating and non-replicating vectors. The nucleic acid vaccine includes DNA and RNA, and the protein-based vaccines include protein subunits or virus-like particles to develop the vaccine. Virus vaccines, at least 7 teams are developing vaccines using the virus itself in a weakened or inactivated form. Many existing vaccines are made in this way, such as those against measles or polio, but they require extensive safety testing. Sinovac Biotech in Beijing has started to test an inactivated version of SARS-CoV-2 in humans. Viral vector vaccines. Around 25 groups say they are working on viral vector vaccines. A virus such as measles or adenovirus is genetically engineered so that it can produce coronavirus proteins in the body. These viruses are weakened so they cannot cause disease. There are 2 types, those that can still replicate within the cells and those that cannot because key genes have been disabled. Nucleic acid vaccines. At least 20 teams are aiming to use genetic instructions in the form of DNA or RNA for a coronavirus protein that prompts an immune response. The nucleic acid is inserted into human cells, which then churn out copies of the virus protein. Most of these vaccines encode the virus's spike protein. Protein-based vaccines. Many researchers want to inject coronavirus proteins directly into the body. Fragments of protein or protein shells that mimic the coronavirus outer coat can be used. This Nature article was authored by Ewan Callaway and is available in Nature Volume 580, pages 576 to 577, published year 2020. Pfizer expects its vaccine to be available in 18 months. The Oxford group says 18 months is too long. The big challenge is compressing a process that can take 10 years into 10 months. It will probably sacrifice safety. The race for the vaccine is on.